Fantastic. So flow cytometry is a fairly ubiquitous technique in biological research labs and in diagnostics labs and in various other industries, so the oil industry. And what I'm going to do, I'm going to go through how it works when we do the first incubation. So if we can sort of keep it fairly synchronized, so you all put it onto your cells onto incubate at the same time, I can then spend 25 minutes or so explaining how it works. But the very basic explanation is it looks at cells one after another, it shines a laser at them, it looks at their size, it looks at how granular they are, and it looks at how much fluorescence they produce. Because fluorochromes, fluorescence, you only get light given off when you put light in. So we put light in with a laser, we get coloured light coming off. And because we're using different antibodies with different fluorochromes, we get different coloured lights for each of the different markers. Okay? And this is, cytometry simply means looking at the properties of cells. So microscopy is a cytometry technique, so is Western blotting. But this is better than, say, Western blotting. Western blotting is a whole population technique. You take your whole well, your whole flask, whatever, and you look at the total protein in that cell pair. Now that's okay if everything's the same. But if you've got a mixed sample, Western blotting can obscure the results, because if you've got some cells that have high, some cells that have low, and you run that through a Western blot, it says that the cells have moderate levels, medium levels. But that's not quite true. And microscopy can give you very detailed information about individual cells. What you can't do is look at large numbers at any one time. The flow cytometer can look at, well, this one won't, but a more modern one will look at maybe 20,000 per second. So if you've got very rare cells, you can still look at 2 million cells, 20 million cells in a fairly short time and look at how much protein they have rather than relying on the entire population. So it's a very, very powerful technique. In diagnostics, it's used to look at HIV status, because you can label your CD4 T cells one colour, your CD8 T cells another colour, and you can see what ratio you get between them. And that's the important thing in HIV. If the ratio's level, the patient's fine. If one of them starts accelerating on the other, that's when the patient will get sick, and you treat with drugs. You can look at leukemias, you can diagnose those by looking at what surface markers are on the blood cells. It's heavily involved in the latest COVID research, and obviously it would be stupid not to take the opportunity to mention that. But it's used in that because you can look at the response to the cells, you can look at cells that express it, you can look at T cells that are specific for it, and so on. So because you can look at one cell by one cell by one cell, but very, very quickly, it's an incredibly potent technique, incredibly potent system. Now this is a relatively old instrument. It doesn't have all of the bells and whistles of a modern one. This one can only look at 4 colours at one time, whereas some of the more modern ones can do 35 colours. This one will only run a few hundred cells per second, whereas the modern ones will run many thousands, and so on. But it works, and it does a good job, and this is the only instrument we have, so this seems what we use. But it works in the same way as the more modern ones. The modern ones are just a bit faster and flashier. We have more coloured lights, and so on and so forth. And are smaller. I will sort of show you, but the laser in this one is this box here. Okay, I'll show you properly. But the laser in this one is in a box about this big that produces loads of heat. Modern lasers are about this big and produce no heat because they're all LEDs. So there have been advances in technology, but this is the technique that is probably the most common technique in a biological lab. More people do flow cytometry probably than any other single technique, simply because you get a huge amount of data with a cell-by-cell analysis. So it's an amazing technique to understand, but the problem you get both with flow cytometry and many other modern techniques is it's all a black box. And you will see this if you're doing a whole range of techniques. You put the cells in on the tube there, You press go, dots come out here. Well, what are those dots? What do they mean? How do you interpret them? And that's part of the problem. And the more modern instruments that can do more colours, they say everything's automated, which is brilliant as a sales technique. And I've been a salesperson for a year or so. It's a great sales technique. Everything's automated. You rock up with your really complex sample, put it on, analyse. Great. But understanding what it is and how it works is really, really important, because without that, how do you know your data is real? How do you know it's correct? And therein are some of the problems. So what we'll do is we'll label our cells up. If we can keep reasonably synchronised, I know it's difficult because we've only got limited centrifuges and various equipment, but we'll get them labelled up. Are you all familiar with the antibodies as well? Do you want me to cover antibodies? People, all right-ish? So the antibodies we're using have a fluorochrome attached to them, a fluorescent protein. The most common fluorescent protein is GFP, green fluorescent protein. That was originally developed from jellyfish. And if you keep an eye on things like New Scientist, you'll see GFP cropping up all the time, because you will see mice that glow green, and you'll see tumours that glow green, and so on. Green fluorescent protein is used by jellyfish. Little bits of light, certain wavelengths, get to depth, and the jellyfish then just glow green. And they use it for signalling and self-defense and all sorts. What we've done is we've taken that GFP, make it in the lab, and we stick it to an antibody. So the antibody recognises a particular sequence on a protein. If it sees that sequence, it sticks very, very tightly to it. If I remember rightly, the antibody-antigen bond is one of the strongest bonds known in biology. So it sticks really tightly. Now, of course, if it sticks tightly and on the other end of it is a fluorescent protein, anything that has that sequence on, that marker on, will stick antibody to it. Anything that has antibody stuck to it will have fluorescence. Okay? And obviously, whereas GFP is green, we now have lots of different fluorochromes. So the ones we're using today are, and I can't remember, I don't know the protocol. Just check. So we're using CFS, which is effectively green fluorochrome. We're using 5-pyrethrin, PE. So 5-pyrethrin is a very bright orange fluorochrome. We're using APC, which is a red fluorochrome, and we're using PerCP, which is a red fluorochrome. Now, the difference is that PerCP and CFS and 5-pyrethrin all glow when you hit them with blue light. So we have a blue laser in the flow cytometer. So when the PE, PerCP and CFS are hit by blue light, they glow green, orange, red. APC, however, isn't affected by blue light. That's affected by red light. So when the red laser hits the APC, that produces red light, far red light. Okay? So we can use all 4 of these together because they're all different colours of light. Yes, because, well, the APC, they are both red, but the red spectrum is quite broad. So you've got red and far red and infrared and so on. So the APC tends to be far red, whereas the PerCP is near red. So as they go through, they're hit by these lasers. They glow 4 different colours in varying amounts, and we look at the amount of each of those colours using 4 different detectors. We can actually measure them very accurately, and we can say there is this much green, this much red, this much orange, this much far red. From that, we can work out how much of each of the different markers we have. Okay? Right, so I think we're almost ready with cells? Okay.
Those markers, so the PE and the
Yeah.
ones you just mentioned, are they typical, or are there more than one?
Oh,
Are
there are thousands of fluorochromes.
So how would you choose which is best one?
Well, there's lots of ways. Sometimes it's availability. So if you want a particular antibody, if you want CD4, again for human T cells, helper T cells, if you open a catalogue and look down the list, you will see 100 different fluorochromes. Because CD4 is very commonly worked on in a whole load of different fields, so people want choices. If you look at some of these markers, it's more difficult because not many people work on them, and so there's far less demand. And so you're not generally doing lots of different experiments, you're doing one sort of experiment with it. So sometimes availability, it could be that the one of your markers only comes in the antibody. And there are rules over, well, there are specific ways you should decide what fluorochromes to use. I haven't really got time to go into it. I normally run a full day theory course of flow cytometry.
No, just curious.
And this takes up about an hour and a half, the choice of fluorochromes. So it is a complex thing. But it could be choice, it could be cost. So some of the new fluorochromes, brilliant violet dyes and things like that, can be £300, £400, £500 for a tube.
I think what I'm getting at is are some better than others?
Yeah, so some are bright, some are dim. And what you also have to think about, that I will cover a bit later, is that when we put the green fluorochrome through, it has an emission spectrum, so it has light that it produces, and it produces mostly green, a little bit of orange, a tiny bit of red. The orange fluorochrome produces loads of orange, a tiny bit of green, and some red. And the PCP produces loads of red, and a tiny bit of and a little bit of orange, and a tiny bit of green. So they do produce light, which we have to adjust for, and I've already done that. But some fluorochromes have an emission spectra that's sort of that shape, doesn't really affect others. Others have an emission spectra that's like that, causes lots of other problems. So there's all sorts of different reasons to choose a particular fluorochrome. And when I say some are better than others, it's very subjective because it depends what you're actually looking at. So if a bright fluorochrome sounds good. Sometimes it's too bright, so sometimes you want a dim fluorochrome. A lot of times you need a bright fluorochrome. So bright is not necessarily good or bad, it's if appropriate. And therein lies the real trick to flow cytometry. Because there are so many different things you could be doing there, so many ways you could be looking at your cells, having that understanding of what the different fluorochrome do and why you use them and which you pick, that's the thing a lot of experienced research scientists still haven't got. And I've trained about 6,000 or so on my theory courses over the past decade. And there are people in there who are professors and have been in the lab for 10 years or 15 years and have been doing cytometry for a lot of time. They come out of it thinking, I might have to go and redesign some of my experiments. Because they just literally open a fridge and pick the nearest antibodies out, throw them on their cells and say, yep, that'll work. And it doesn't always work. You've got to actually think about what you're doing. It sounds really patronising to say to a room full of research scientists with years of experience, look, you've really got to think about your experiments. I get some very strange looks, which is why it's better doing remotely, so they can't give you the strange looks. But it sounds patronising, so you've got to think about what you're doing. And actually, my full-day theory course gives a basic introduction. I've been doing this 26 years, and I'm still learning things about flow cytometry. So there's all sorts of facets to it, and obviously we can't cover the vast majority. Yes. So you've got all. I mean, this has no real automation, both in the software and on the instrument. But one of the instruments that I used to be the application specialist for will run a 386 well plate in under 10 minutes through a flow cytometer, and a 96 well plate in under 3 minutes. And a drug company I know of in Slough, they have one of these instruments set up with a plate loading robot. So it lifts plates out of a tray, out of a rack, and loads them onto the instrument. And it runs them, and then the robot takes the plate away and puts a pressure plate on. So it's running all day, every day, every 3 minutes, well, every 5 with a wash, every 5 minutes it's doing a 96 watt plate. In the middle of the room, they have a robot that's as big as this bench, and they load it up with antibodies at this end, they put the tubes of cells on at this end, empty plates in that corner, they program it all and press go, and it pipettes cells, it pipettes antibodies, it has plate centrifuges, so it spins them. So what you're doing now, it does that on 96 watt plates all day. And the robot just runs it. And it outputs a rack of plates at the end. And a technician comes along and lifts them off, puts them on the plate loading robot, and they're loaded into the cytometer. And they empty the waste bins on top of the fluid. And so it can run hundreds of thousands of samples a day, every day, on this. And it has heat map software. So it looks at the 96 wells, looks at the colours, where the software has graded it low, medium, high, very high, whatever. It assesses what ones to work at, and then it alerts the scientists and says, right, you need to look in more detail at this one, and this one, and this one, and this one. And it's brilliant. It's great. And now, I mean, all right, you've got about £1.5 million worth of kit there. But that department within the drug company is a screening department. What they want to do is they want to look at hundreds of thousands of drugs and drug combinations per day. So they want to treat the cells, and they want to do a dosing scale. But when they do a dosing scale, they don't want to just do 1, 5, 10, 50. They want to do 0.1, 0.15, 0.17, 0.2, 0.21, and so on. So they might run 10 plates just to do a curve, and so on. So for them, it's brilliant. Now in a research lab, you'd never use it because you tend to be running experiments that have 5 or 8 or 10 tubes in. So for those, the lesser automation of the other systems where you can have a tube rack. So one of the instruments I've been sort of demoing to the staff here to potentially buy, you can put a 24 tube rack on, click go, and it just read all 24 tubes. So what we would be able to do with that is you'd be able to run all 3 samples each tube, we'd put all 15 tubes on, I'd click go, and then I'd stand back and talk to you while it's running all 15 tubes. Then it would output all of your data in a standard template format, and I'd click print, and we'd have PDFs, and I could hand them out to you, and it'd have all the analysis done while I was talking, which is nice. They're not totally free from error, and in a way, the increased automation is a problem. Because if you get used to putting it on and clicking go, and then your data comes out, and then you click go, and your data comes out, you're not used to looking at the data and seeing if it's correct or not. And actually, diagnostics labs, hospital labs, are finding this problem more and more. Because all the companies that sell the instruments, they look at all this wonderful automation, look how easy it is. You don't need trained staff to run this, anyone can do it, until there's a problem. And then, because the hospitals then get rid of all the senior staff who know how to operate the things, and have technicians who are effectively just refilling and emptying, it means that when there is a problem, the instrument is misdiagnosed, a leukemia or something, there's no one around to actually spot that it's done that. And then you start getting errors. And then the people who run the diagnostics stuff say, well, why didn't anyone spot this? You know, they say, well, you've got rid of the people who could spot it.
So how would you pick out that misdiagnosis?
Well, again, it's experience. So you see, so what it'll do is it'll produce the data like you'll see it in the paper there, you'll see it on the final page, the docs and histograms and so on. Now I can look at a page of data and see if it looks right. I get at a glance, because I've been doing this so many years. An experienced cytometrist will be able to look at CD4, CD8 and go, yeah, that's okay. Literally in about 2 or 3 seconds, because it'll look right. And so they can look at it, look at the number, yeah, or the yes, they're okay, or no, they're not, from the software, and they will know immediately that that's okay. But if there's a problem, so there's a blockage, or there weren't enough cells, or the blood was contaminated, or something, they'll pick it up and go, no, we're going to have to redo this one.
Software work that out?
Not always. Not always. So the software is quite good at spotting problems, but it's certainly not infallible. The human brain is much better. Flow cytometry, when it comes to data, is very much a pattern recognition thing. So your brain looks at it. It's like, you know, those magic eye pictures where you look at it and you see a tiger, or you don't. In a way, it's the same with flow cytometry. If I look at the data, I can see the tiger there that says it's wrong. And my brain may not even know what specific things are wrong about it. It'll just say that's not right. And then I'll look into it and say, now, yeah, okay, so I see that one's and the gate there is wrong, and so on. But it's very much, it's really hard to explain, but it does come down to experience. And just
So it's a visual thing.
Very much a visual thing.
Even though it's made up of data points, it's visual.
Yes.
I would have thought
Because
that.
it shapes.
Yeah.
So if you run a complex, say, blood through, you get one population here that are your neutrophils, and one population here that are your monocytes, your lymphocytes, your platelets, your erythrocytes. And I'm used to seeing them in a particular orientation. So I know what's supposed to be there. And I was reading a paper the other day, and they appeared to have no neutrophils or granulocytes at all. But the paper didn't comment on it. So I'm reading it thinking, someone somewhere is messed up, because that is not whole blood. That can't be whole blood. I do not believe that this patient has no neutrophils, granulocytes, or whatever, and it isn't remarked somewhere. You do get patients like that, but that's quite a noteworthy thing to put in the paper. You don't say, we took blood from our normal donor and it showed this. No, it didn't. But I see that because my brain says, this is what I tend to see. So it is getting used to it. And so, obviously, given all this complexity, it's quite a challenge for you to learn all of this in a couple of hours and then analyse the data in a couple of hours. So I will send through a handout detailing some of the basics of flow cytometry for you after today. And I will also go through in the 2 half-hour incubations just how we actually look at our data and what the plots mean and what the scales mean and so on and so forth. Okay? Good. Right. I think we're...
Okay,
Are we
so
ready?
and here we are ready. So we have done the first step on the experiment. So what we think that we need to do now, we have all the samples. What we're going to do is just centrifugation the samples, all of them. And after, what you need to do is starting in step 2, where you would suspend them already with 500 microliters of fixation buffer. Okay? So we have done the cell counting for you. That is sorted. We will just need to do calculations after when we have your report, okay? But we will do those after. So, alright. So what we did at the moment is we have the cells in suspension, and we were just making sure that we had enough cells for you to start the experiment, considering that the minimum cells required are 5 times 10 5. There you go. Otherwise, we get with what happens to this if you not know about it?
When I did my master's degree, because I did my master's and PhD down at Essex University, and my master's supervisor was an advisor to one of the big centrifuge companies. And he showed me some pictures of a place he'd been called out to. And it was a spin-out centrifuge. So you have metal buckets that screw into a cap, and then you have hooks, and they hook onto little pins. So as the rotor spins, the bucket swings out on the hooks. And these are ultra centrifuges. These run sort of up to 1 million G. So they're ridiculously high speed. And what happened was someone had hooked the bucket on, but rather than hooking them over the pins so it could swing out, they managed to hook it onto the top of the pin. So as it started to speed up, the bucket actually the hook crept round the pin. but it was just on like that. And it got up to speed. And then there was a slight vibration, and it came off. Now these centrifuges are lined with titanium walls. And the centrifuge bucket punched a hole in the titanium, punched a hole in the breeze block wall of the centrifuge room, punched a hole in the wall of the other side of the corridor, travelled across the office, punched a hole in the breeze block wall of the office onto the outside, and disappeared off into the outside. And I think it was only found about 6 months later because it just went, half a mile or whatever it was. Meanwhile, of course, the centrifuge is now not balanced. Now, if you don't balance this one, as you say, it jumps around and shudders. This centrifuge, without its missing bucket, because it was spinning so fast, leapt around the room, destroyed just about every other instrument in there, and destroyed itself. And it did about £3 million worth of damage to the instrumentation in there.
And did it get hurt?
No, luckily no one was in there. Had they have been, it would have been very, very nasty. But luckily no one was hurt. But my supervisor showed me all of this and said, right, now we're going to use the centrifuge with these swing out rotors. And I was like, I'll let you do this bit. So it didn't quite get it the right way around. What he should have done is show me how to use it. And then when I've used it successfully, he said, but don't get it wrong because.
Yeah.
So
Did you do it right?
I did, and you know what? Every single time, I always ran it. I'd hook the buckets on, I'd swing them out, and I'd check them, and I'd tap them, and I'd rotate it. I'd close the lid, and I'd go, right, and I'd open the lid, and I'd check the buckets. Which I was probably overcautious, but actually, in a way, making mistakes is good in research. With cytometers, when I'm teaching people hands-on, there's one type of cytometer where it draws up loads of air if you don't put your tube in at a particular point. And I always say to people when I'm running the course, when it says this prompt, put your tube in, then click OK, don't click OK, then put your tube in. And then we'll go through it and say, right, you load some sample, and then click on the thing, and the prompt will come up, and then click OK, and I'd stand there going, yeah, right, OK. And I'd say, right, what have we forgotten? And someone else would go, then put the tube in. Yep, exactly. So now it's full of air. So now we're going to have to purge it, and I'd use it as an excuse. But you could bet your life that that person, with the embarrassment of missing that step, never does it again. And so when it's safe to make a mistake, it's good to actually make mistakes in research. And any researcher who tells you they've never made a mistake anywhere is lying, because they have. I've done things like I made a buffer up for this that had DSA in it, bovine serum albumin, and I needed it sterile. So I autoclaved it. And of course, what happens if you autoclave albumin? You get poached egg.
Yes.
So you have the nice swirl, and of course, at the autoclave, because it rotates, you get this swirl of poached egg. So I left that on the shelf on my bench for all the time as a postdoc to say, don't be an idiot. So if you're at the start of sort of your research career, you will make mistakes. You will. But you've got to learn from those and not make them again.
You could have a new discovery.
Well, absolutely. There are times when you put the wrong thing in, you found something out you didn't even expect to look at, and I've done that as well. So then you also get bad practice, such as a PhD student who kept leaving my antibodies on the bright windowsill. And the thing about fluorochrome is, if you hit it with daylight, it keeps fluorescing, but that wears it out. So you end up with a non-fluorescent fluorochrome. It's called photobleaching. If you leave a fluorochrome, so I've got fluorescent beads in here. I mean, they've only been in there for 20 minutes, it's not a problem. But if I left these out for a few days, they wouldn't be fluorescent anymore. And if I left them in really bright sunshine, by tomorrow they wouldn't be fluorescent anymore. So that's why all of the antibodies are in boxes. Very often, I mean, the beads in that tube come in this foil packet to block out the light. So when you come in and you find your 3 really expensive antibodies in a rack on the windowsill in clear tubes, you tend to get a bit cross. That's not a mistake. That's being stupid. So. Right, are we ready to?
Yes, we are. So we have the cell. So you need to be really careful with that because you might almost not see the cell pellet because the number of cells are really reduced. So the pellet of cell will be on the bottom of the tube, very kind of milliliter in here. So what you need to do is to remove gently the PPS, because the cells are in PPS. So the cells will be really attached to the bottom of the tube. So make sure that you remove the PPS gently without touching the cells, the surface of the bottom. You can leave a tiny, very tiny, tiny bit just to make sure that you are not aspirating the cells out.
Yes, better to leave a little bit of PBS behind
Exactly.
than take too much. If you take too much, you take the cells,
Exactly.
and then you have nothing in the end.
Okay. So aspirate all the PBS first. Use one different tip for each one of the samples because we have different cells. Okay. Discard all of it, and after I will have in here the fixation and the fixation buffer that you will need to add 500 microliters to each one of your tubes. Okay? Because we just have it in here, so we'll need to make sure that all will go around and we take 1 milliliter. In there is 1 milliliter in total. So if you use the 1 milliliter PET tip and set it up for 1 milliliter, okay, after you are able to aspirate. Be careful when you remember first the finger in, outside, okay? So you have your PET outside, just press to the first stop. Inside, gently release, because if you go inside and press, there will be bubbles going in and your cells will be disturbed, and after when you remove PBS, there will be cells going out. Okay? So, first outside, press it. Inside, release gently, and dispose. Yeah? Questions?
There we go.
This remove, this should be removed, and
So this is always strange for me when I'm teaching people this, because I've been working in the lab.
yes, that's not good. I mean,
It's much easier to pick the tube up and hold it up to the light. So
one heavy hand, yes.
it's also a good idea to tip the tube slightly, I find.
We're outside, first stop, go inside, gently see where you don't touch the bottom, touch the side walls of the tube, preferably. Okay. You want to bring it to the level of your eyes, it makes it easier. Leave just a little drop inside to make sure that the cells are not linked.
When you say pellet, it's going to literally be stuck.
If you look, you should be able to see a little white.
You can see just a tiny, tiny bit.
Yeah, this is
Very, very tiny. See where we are. Yeah, that's fine. That's okay. We like that. Yeah, that's fine. We like that. Yeah, we want to go.
Yeah.
Bit more, just a little bit up here.
Yeah.
Okay? Then just leave it a little bit. Good.
Yeah.
Very good. Technique. That's it.
It's well, it's up to you. I mean, if you'd rather do it when the experiment's finished.
Well, I can do that's the thing. I just don't want to get away with it.
It's a good problem. Sometimes it does very well. 1994.
It doesn't take
No. It becomes
it's exactly the
Once we're used to it,
yes.
but the first time, it's not very like
Yeah.
so that. So second
It's always quite stressful this stage, because when you're doing a research experiment and you've got these really rare critical cells, you've only got one lot of them, and you can't even sequel it, and you're removing the supernatant, and you're thinking, if I go too far, I get no data whatsoever, and the whole bit of this project is ruined. So it's, yeah.
Well, we need to be positive because we have five people who do the same thing. So hopefully one of them will get it right.
Having said that, with practice, so I actually tested myself once. I used a cell counter, and I tested it at each stage to work out what I'd lost. And I think I got my losses down to about 3% over multiple washes and so on. So, you know, once you get confidence as well is really important here. If you're really, really nervous about losing your cells, you tend to lose more. If you're confident, you tend to have less problems. So, right, so the fixative we're adding now is it PFA?
Now one is session buffer. I think
It may not even say.
it
So I think it's probably PFA, paraformaldehyde. What this does is it cross-links the proteins on the cell surface. So it forms a scaffold between all the cell surface proteins. That then makes the cell very rigid, very solid, and it stops it falling apart because the other component of this buffer is a permeabilization agent. Does it say what that is?
It's from
Yeah. Does it say what, is there a well, it's a methanol. So methanol is our permeabilizing agent. So a permeabilizing agent knocks holes in the cell membrane. Now, if you did that without fixing it, you would actually just dissolve your cells. They'd all fall apart. So it fixes it and makes them rigid, and then removes chunks of membrane, so that some of the antibodies can then get inside to label inside the cell as well as outside. Okay? This is not a step you run for every. Yeah, one example. Oh, sorry, it's got this one's got saponin. So saponin is a plant-derived detergent, and it dissolves the membrane away, so it allows the antibodies inside. It's better than methanol.
But because they've been fixed.
Because they've been fixed, they won't fall apart.
Right, but are they just been debris?
No, because the fixative actually ends up getting into the cell, so it cross-links the whole cell, makes all the proteins rigid, and then the saponin dissolves away little chunks of membrane. So these then act as, these are big pores on the cell surface that will allow the antibody in, but the cell is solid, effectively. And I once ran some demo cells on an instrument, one that actually takes images of the cells, it's supposed to take images. And I ran this demo sample, it was 10 years old. The cells had been fixed 10 years previously, and they looked as good as the day they were first produced. So in theory, with fixing, you can leave cells quite some time.
Now, fixation.
Yeah.
Would you not want to do that if you were decentralized it, so then you can maintain the structure?
Yeah, so for some things you don't want to fix. Because firstly, fixing kills the cells. You can't look at live dead status, for example. The other problem with fixing is because you cross-link your proteins, some markers that you might be looking at end up being cross-linked, so no longer recognised by the antibody. So fixing can stop some antibodies recognising your markers. So it's one of these things that when you're designing your experiment, fixing, non-fixing is like the antibody choice. There's all sorts of different stages that sometimes you do, sometimes you don't. And there are different fixatives. And there are different permeabilization agents. And so how long is the incubation for? when everyone's got their cells fixing, then we will gather around the cytometer and we will have a look at how it works.
Is it really between 2 and 8 degrees?
No, so I ideally you put them on ice. To be honest, do you want to put them on ice or should we do? Yeah. Yeah, so we would normally put them in an ice bucket. The reason is, what you do with the vortex,
You can put it on it.
you can touch it.
That was really scary.
You just touch it in the end, but it's not. It's quite sensitive. So.
My goodness gracious.
Now, I will show you something. You may or may not be able to get this. Okay? I'll show this to everyone, and it's one of these things I've rolled in your tongue, certainly depends on the pump. But I'm not a huge fan of vortexes. They can be quite harsh on the cells. They can break up some of the cells. And if you're working with small samples, small cell numbers, you don't want to break up your reactor. So to resuspend them, I'll just take one of your tubes. The way I like doing it, you hold the tube, and you flick things across the bottom. Okay? And that works. You get really good mixing, and then you just the bubbles away. It will, because it's frothy, because there's something in it, which is a detergent. There's about 9% detergent. So effectively, it's soap and water, so great. So that's good, excellent. Try using all of the fingers though. And yeah, and it's one of those things you can do. Brilliant, that's fantastic. Have you done it before? No? It's really weird. Some people can, some people can't. Different techniques. There's lots that work. I've always found that just running the fingers across the bottom agitates it really, really well without damaging the cells too much. So, have you labelled your tubes with your initials and what they are? Always label your tubes. I've been in far too many research labs where you open a fridge and you see a whole rack of tubes labelled 1, 2, 3, 4, 5 up to 65 or whatever. No name, no date, no ID, nothing.
Someone's going to have to go and make this one. Bloody four corners. No, that's fine. We'll over it.
Well, as long as they're all labelled, that's the thing, it's not where we go. Yeah, I mean, we do video these and it can be useful, but it's not really a replacement for actually seeing it in person. You know, and I've been doing a lot of remote courses recently. And remote theory is fine, remote software is fine, but any sort of hands-on cytometry, you really need to be there. So.
Want to do a course and how long is it?
Well, I mean, I do full-day theory courses, and that gives people the basics. I have in the past run full-week courses where we do a mixture of hands-on and so on. And even those, they will give you a really good grounding. They're not going to make you an expert. That's experience. So training can be anywhere from, you know, let's say an hour for a really basic intro up to a week. So, well, when I run my theory courses, the cost per trainee generally works out to be about £40 if I do it remotely, which is not a huge amount. But you see, I have got like a cut down half-day course, which I have run for an MSc in Bradford. And that, if there were 10 trainees, would be £20. If there's only 5 trainees, it would be £40. But it's still not a huge amount to get a more in-depth understanding of the subject. So, I mean, that's certainly discussable. So, right, have we got all of the...
So everything is in here now, so they're invited for half an hour.
Good. So what we'll do, we'll come around here and we'll see the instrument itself, yeah? Is that okay?
Just try to kind of not clump all together with
Yes.
health and safety, all of that, so we just need to kind of keep the distance as much as possible, so I'll keep the lights.
What we need is like one of those school experiments where, you know, you stand in the tray of detergent, lift the ring up and you get the bubble around you. That would be quite
Yeah.
good.
did that at our secondary school.
Yes.
Yeah. At our secondary school, my teacher got, this was in Hadley, he got a pair of cow's lungs from the local abattoir and he inflated them, and when he pushed on them, it moved because it still had the larynx intact. And I still remember that all these years later. But that's science, it's, if you remember it. So, here we have the flow cytometer. There's not a lot on the outside. We have our standard flow cytometry tube, just little polystyrene tubes. And they go onto a little metal needle that takes up our sample. And then we've got some control buttons. That's about it. And actually our control buttons are very, very simple. We've got one that's low, medium and high. That's how fast the cells run through the system. We've got one button that means it's running. But while it's running, fluid is now running through. So fluid is running from here, which is just water. Fluid is running from here. But it's just running, and it's pointless leaving that on. Added to which, our laser is on as well. And lasers are like light bulbs. They have a lifespan. You don't know what it is, but when it reaches a certain number of hours, that laser will just pack up. So when you put it in standby, it switches the laser off. And then we've got prime mode, and all that does, it forces some air through and then some fluid through to get rid of any potential air inside the system. Now, as far as what it looks like inside, here we have our laser. Okay, so this is our blue laser here. This is our red laser. So because this is an LED laser, these were added later on in the lifespan of the instrument. This is our red laser. And the laser light is shone all the way through here into the bit behind here. Now you can't really see because of the tinted glass. But what there is in there is a little chamber shaped like that. Because we want to look at one cell after another. So what we do is we feed our cells into that shape, and we feed sheath fluid. We feed PBS, which is in a tank here, around the outside of that V shape. So fluid dynamics says that if you have two streams, but you haven't got turbulence, they don't mix. Okay? And you can actually see demos of this on the internet, where you have, you know, an angled board and you run red coloured water down in blue coloured water, and as long as you've got a really smooth surface, you get a red and a blue line of water coming down. They don't mix together. If you then put some little crenulations or whatever, all of a sudden they mix, and you get sort of dirty brown colour as all the colours mix. So the sample going up through that V shape doesn't mix with the PBS coming in around the outside. And it all gets squashed together as it goes through the V, until it gets to a very, very narrow bit at the top. And then it goes through a sort of a straight chamber. And because everything's been squeezed, your sample has been squashed down to about 10 microns wide. And it's surrounded by a column of sheath fluid. And that's why it's called sheath fluid, because it forms a sheath all around it. So we've got a stream of cells 10 microns wide. So what happens then is, because your cells are around about 8 to 10 microns, you've got one cell, and then another cell, and then another cell, and another cell, going through that narrow chamber. Then the lasers come in and hit them. So as the blue laser hits the cell, any fluorochromes excited by blue light start to glow. And what we have here are detectors. So we've got a green fluorochrome. So blue light hits the cell, that green fluorochrome starts to give off green light. The green light goes through a whole load of different filters here, multiple filters, and is picked up by a detector, a green detector. And it says there is that much green light, and that goes down the wires of the electronics. Orange light is picked up by another one, red light by another one, and far red light by another one. What it also does is, when the blue light hits the cell, anything really, really, really small in the cell will scatter light. Not all of it, the vast majority of the blue light goes straight through, but some light will be sent in all directions, some blue light. And we measure that here, and that's what's called side scatter. It actually goes in all directions, but we measure it to the side. And the more granular a cell is, the more it will scatter light in all directions, so the more we'll pick up here. So if you've got something like an erythrocyte, a red blood cell, that's a bag of protein. That's all it is. There's nothing really granular in there. So the blue light goes straight through and nothing's scattered. You've got something like an eosinophil, a granulocyte, a neutrophil, something like that. That's full of organelles and granules and all sorts. They scatter light in all directions, we pick up a lot. So a neutrophil has a high granularity, an erythrocyte has a low granularity. Now, a cell is too big to scatter light in all directions. What the cell will do is it will bend light forwards. So the blue light's coming through, when it hits the cell, some of the blue light does that. Okay? And that's called forward scatter. So it's just being bent over an angle of a few degrees. And we have a detector here to measure that forward scatter. And in general, the bigger a cell is, the more light we pick up in this detector. Okay? It's not an absolute relationship, but it's reasonably good. So what we can tell about every cell is when it is hit by the laser, we know for that cell how big it is, how granular it is, how much green fluorescence, orange fluorescence, red fluorescence, far red fluorescence it has for that one cell. That data is stored for that single cell. We then move on to the next cell that is moving through. And they end up coming through the flow cell here, and they come out this tube, and they go through, and they go into a waste tank in here, okay? And so all of this electronics, all of this fluidics in the drawer, and there's fluidics in here, and there's pumps, and there's valves, and so on, all of that is designed to get the cells into a single stream so you can hit them with a laser and measure their fluorescence and other properties. Now, interestingly, the forward and side scatter thing is also why the sky is blue. And although it's not strictly cytometry, it might win you a pub quiz one day or something like that. Because, or you know, if you have children or if you have children in the future, you're going to get asked why is the sky blue, because it's one of those things. Well, we've got 500 nanometer particles in the upper atmosphere. So as the mixed wavelengths of light come in, any wavelengths that are bigger than those particles are side scattered. So the 488 nanometer light, our blue laser light, the particles in the cell are smaller than 488 nanometers, so they scatter light in all directions. So the longer wavelengths of light, so the reds, the oranges, and greens, and so on, are scattered in all directions. And some of that goes back out in space. And some of it goes in all directions down here. But because if the wavelength of the light is smaller than the particle, so the 488 nanometer light, 488 nanometers is smaller than our cells. So the 488 nanometer light hits the cells and is bent. And so our blue and violet and ultraviolet light hits the 500 nanometer particles and is bent forwards and comes down to us. And because we get more blue light than other colours, the sky looks blue. Now, if you've got, of an evening or a dawn, the right particles in the right place and a long light path, you get enough red, orange, yellow light scattered, and therefore you get the red and sunsets and dawns. So that's why the sky is blue. Now, it isn't cytometry, but it's related to it. It's a good way of remembering the difference between forward and side scattering. If the particle is bigger in the wavelength, forward scatters. If it's smaller, side scatters. Okay? And so that is all set up to run these cells at hundreds of cells per second. So hundreds of cells per second are fed up from the tube, through the laser, through to the waste. That data is all taken off, processed by these rather, these days, primitive electronics. Nowadays this will be a computer chip having all of this rather than all of these big circuit boards, but it works. And that data is sent to the computer. How are we doing for time? Have we got, what's the timer say? So 30 minutes, okay, so we've got a bit of time now. So you need software to interpret this data, because of course what you're getting is several hundred data points per second, each one telling you the forward scatter, the side scatter, and the fluorescence properties of that cell. So if I click on Run Now, all we will get at the moment is debris coming through. So you can see there's some dots appearing here. Not particularly quickly, but they are appearing. And we're looking at something like 60, 70 events every second. So when something goes through the laser, that is referred to as an event. Now, normally, if we put a tube of cells on it, will be cells. But not every single event that goes through is a cell. If I put on my QC tube, these are beads, but they still come through as events. So you've got to remember that not every dot you see will be a cell. But each one of these dots, you can't really zoom, you can zoom in on this and see if this works. So each one of these dots is an individual event. So this is just a tube of water. So what we're seeing here are tiny flakes of plastic maybe from the tube. It could be tiny bits of dust that have settled in there. It could be micro bubbles, because when you squeeze the sheath and the sample fluid to get them into that narrow channel, you apply pressure to them. And if you have water that has air dissolved in it, as you pressurise it, that air starts to come out as bubbles. They're only tiny, they're only might be a micron across, but they will still trigger the laser. Okay? So this is size, and this is granularity. So forward scatter is abbreviated to FSC, forward scatter. Side scatter is SSC. So this event here is relatively large. This event here is very granular. This is very, very small and non-granular, and so on. Okay?
So when you are collecting your data, then this type of graphs will be generated. For your report that you need to produce, you need to make sure that you understand these parameters and what they mean in order to do your data presentation and data discussion. So it's quite relevant that you get these things right.
Yeah, absolutely. The interpretation of these is critical. And what we'll do is, in the antibody incubation stage, we'll look more at what the data looks like on the plot and what it means. But you see, we can look at forward and side scatter. We can also look at some of the fluorochromes. Now, obviously, we don't have fluorochromes in here. What we're now looking at is FL1, fluorochrome 1, versus fluorochrome 2. So fluorochrome 1 is our green detector. So this is any green fluorescence. This is fluorochrome 2. This is orange fluorescence. So this is where we will see our, is it CLS or something? This is where we see our PE. Now we are getting a little bit of fluorescence coming through. Now this could be what's called carryover. So I ran some beads before. If some of the beads stick slightly on the inside of the tube, they will come through. Now we're only seeing one every few seconds. Now if you were running your cells, and you were running 300 or 400 cells per second coming through, and you get one carryover bead every 3 seconds, it's only going to be 1 in 1,000. It's not going to make any difference to your data. But if we will, and this is where the automation that I mentioned earlier, the superfast plate reader, that's where it starts to fall down. Because it reads by going into that plate, into that well, and 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 you get carryover. And it might only be 2%, and it's fine for a screening system, but you wouldn't want 2% carryover in a research project. Because if you're looking at tuber T cells, and then tuber B cells, and then when you run your B cells, 2% of them are T cells, that's not good. That's not, you can't tell anything. But even though they probably are carried, you don't know for sure, and you've got all sorts of issues. So what we often do is we often wash the probe, we often run a tube of water between samples, and carryover is a problem. But it depends on the cell type. Some cells are really, really sticky. And if they then stick to the tube and then come off, you get a lot of carryover. Some cells are non-sticky. It's also possible to block this. One of the problems we had, I can't remember if it's last year or the year before, was the cells were very clumpy. And we'd resuspend them, and you would hold up to the light, and you'd see lumps floating around. And so if they're visible to the eye. Because the channel, the narrow channel in here, let's just stop this, because the narrow channel in here is only a couple of 100 microns wide. If you get a 400 micron wide lump of cells, it just wedges itself in that channel. Now, it is possible to unblock it. However, to unblock it, you need something like this, and that's actually what these are designed for. Oh, that's seized on, so it's not coming off, and that's seized on too. If you look through there, you might just be able to see there's some whites. Yeah? They're incredibly fine. You might just about be able to see them in there. And what you have to do is you have to feed one of those up the middle of this metal and then into the flow cell to actually physically clear the blockage. It's not an easy job. It's not the sort of job that I want to have to be doing. So when you resuspend your cells, make sure you resuspend them properly. Now what some people do, they put them through a tube top filter. Now these tubes actually, if you look in there, you'll see there's a little mesh. Yeah. So these tubes, these are flow cytometry tubes, but they have a, let's say what the strain is. So it's called a cell strainer cap. So you can actually take the sample onto the top there, tap it through, and any big lumps are held in the mesh. You can also buy separate tube top filters. This is a Celtrix one. That's a 60 micron. So the grid in there will let the cells through for 60 microns or less. So you're not going to block it. However, what I would say is, from a data point of view, and this is for your future careers rather than for today, from a data point of view, be careful. Say we're doing an experiment where we're trying to introduce these pluripotent stem cells. We've got our cells growing in cell culture, we add treatments, we're adding experimental drugs, so on. We harvest them off, and you often harvest them off with trypsin, which is an enzyme that just chops through the extracellular matrix. Harvest them off, spin them down, label them, put them through a cell strainer, one of those, run them on here, and we find we've got very few pluripotent stem cells. You go, oh, the drug hasn't worked. You know, we spent millions developing this drug, it hasn't worked. Oh no. But what if your drug, in addition to making them pluripotent stem cells, also makes them really sticky? They then stick together during your labeling process. You put them through here, all of your stem cells are in here. You put the tube on there, and you're putting through the non-stem cells. So of course you're going to have very, very few of them. So all of a sudden, your data is wrong. Of course it could be the other way around. What if your drug makes them very non-sticky, but normally they are? So you run your tube through, 98.99%. Yay, it's worked brilliantly, I'm going to be a multi-millionaire. You know, we've got this new drug, makes stem cells. Except that what you've done is you've taken a 10% stem cell population, 90% of them are clumped together, and you've pulled them out from the filter. And what you've got left is 98.99%, but that's not a true reflection of your actual data. And I say this because experiments are complicated, so, fluorochrome choice and so on and so forth. And you have to think about each of the stages you are doing, what you're doing, and why. And in a way, when you're given a protocol like this, it's more difficult, because you are told, by the experienced scientists running this practical, you are told, this is what you will do. Why are you doing each of the steps? And you have to know that. And what I would say to you is, even in this sort of thing, feel free to say why. Why do we do this? Why do we fix? Why do we permeabilize? Why do we incubate 30 minutes? Why do we incubate on ice? Etc.
Can you disperse the cells?
You can. I mean, you can sometimes use a pipette to break up clumps, but that can sometimes break up cells as well. You can draw it through a needle. That will sometimes work. You can also add 5 millimolar EDTA, which is a chelating agent that can help stop them being sticky too. But EDTA can interfere with some assays. Pipetting them up and down can interfere with live-dead assays. So my standard answer is it depends. And very often there is no hard and fast yes, no. Very often it's, well, depends on what else you're doing. But I would certainly say, and this is not just cytometry, always, always, always ask why you're doing a particular step. Because if the person running the experiment can't explain to you why you're doing it, do you really need to do it? That's what I would be asking. So, each of the steps in your protocol, I can tell you exactly why we're doing it and what would happen if we didn't. So, are we about done for first incubation? Right, okay, so if you want to carry on with your labelling process, and then we'll look at data in the second incubation.
So you can see what we do next then. Time. And get more paper around it.
So,
Sorry?
yes.
Yes. Yeah.
You could do multiple assays like what's
Yeah.
to
Yeah, because you often want to validate your assay. Yeah. Yeah. But it's, I mean, you see, what you then do is you'd probably, if clumping was a potential issue, bring in microscopy. Because microscopy may well only look at some cells. If you look down the microscope, you see clump, individual cell. You think, right, I've got a problem. Now, can you fix it by EDTA or pipetting or whatever, or vortexing, or not? If you can't fix it, you have to use another technique. If you can fix it, you can use cytometry, but can you fix it without affecting your data? And this is what comes in from your years of experience and training. You have to apply different techniques. But no, always ask for stages. Unfortunately, you see a lot of modern PhD students nowadays, because there are so many kits out there, they buy the kit, they run the protocol in the kit, and the data has to be right, doesn't it? Because they run the protocol. But when I was in the lab as a PhD student, and generally as a postdoc, there weren't many kits. You developed your own assays with your own reagents you bought. So you had to prep everything. You knew why you did every step because you invented it. If you're given a predetermined protocol, it's harder to know why you're doing it because you're just told to do this. So, and there are PhD students who send me data post-operatively to look at it. Labelling's not right. Well, I follow the protocol. So, did you optimise it? Did you check everything was right for yourselves? No, but you follow the protocol. And I think you need to actually think. And it sounds really stupid when it's like that. Yeah. And the problem is time. When you're a PhD student, you've got 3 years to get your thesis. That's it. You've got that deadline, and even when you first start, it's approaching fast. So you don't want to faff around prepping stuff, but on the other hand, you don't understand what each individual step is, your data. So, it's, oh yeah.
We have that. We can take, I can take this from here. So that's fine. That's okay. So from here now, I'm just waiting for isotype controls. Is that okay? So you will add the isotypes exactly the same volume, but you just add isotype controls because it will add you, we're just talking about it, and will add you another control to your sample. Although you have already a positive and negative control cell line, if you have the isotopes also as controls, you can have more bases to actually discuss your data.
Yes.
Yes.
So the problem is cells are sticky. If you just add antibody, if we added these antibodies to mouse lymphocytes, we would get some binding, or we would get some fluorescence, because cells are just slightly sticky. And if you add an antibody, even if it's not specific, some will just stick to the surface. So cells will be slightly green, slightly red, slightly orange, slightly violet, just because some antibodies stick to the surface. Now the problem we've got is we don't know how generally sticky our cells are. They might be really sticky, they might be not sticky. More of a problem is we're looking at 3 different cell types. What if one of your cell types is really sticky and one is not sticky at all? The one that's really sticky would bind loads of antibody anyway and would be quite bright. And we're trying to work out what's got our markers on it. We don't care about the general stickiness, we want to look at our markers. So what we do is we add an isotype matched control. So antibody isotypes are IgA, IgE, IgM, IgD, and IgG. And they're actually subtypes as well, so there's IgG1, 2A, 2B, 3, and so on. And different isotypes have a slightly different structure. So IgGM is 5 antibodies arranged like in a ring. IgGD, let's go back to the years, I think it's a dimer with a binding chain and so on. And all of these will have a different natural stickiness themselves, so they will stick at different rates to different cells. So what we have is we have an antibody. If, say, our OCT4, say it's a mouse antibody, and it's an IgG1, then we will have another mouse IgG1 that doesn't bind to anything on human cells. So it has no binding at all to human cells whatsoever. But it's still a mouse IgG1, and we put the same fluorochrome as the OCT4 antibody. So that's our isotype matched control. So it's the same isotype, same animal, same fluorochrome as our actual antibody. Those of you who are doing the isotype matched control, when you add that in, you will get some natural stickiness, but no specific binding. So you will incubate, you'll run it on here, and when we look, we will use that to say, and we'll run those first, and we'll say, right, this indicates no specific binding, this peak here. When we run the other 3 sets of samples that have the actual antibody, any shift to the right of our no specific binding peak indicates actual binding. It has to be. Okay? Are you all right with that? Yeah, it is. It's calibrating our experiment. Now, it's a daily thing. So if we were to run this again next week, we would have to run the isotype matched controls again, because it could be that one of the cell types goes really sticky when the CO2 in the incubator drops to 2%. Now, it could be over the weekend. There's a problem with the CO2 cylinder, and for half a day it's at 2%. So one of our cell lines becomes sticky. We don't know that. So we would have to run these isotype matched controls every time. Because then, if our cells 1 week are really sticky, it doesn't matter because we're still setting our baseline on the background stickiness. Okay? Whatever it might be.
Did you say the isotope control is less sticky than the
No, so the isotype matched control should be exactly the same stickiness as our actual antibody. That's why we use
the different peaks?
So, well, when we run, we'll see it when we run them, but when we run our isotype matched control, we'll get AP, say there is this much fluorescence. When we run our actual antibody, If it's in the same place, we have none of that on that cell. But if we run it to the right, if it moves to the right, it means there is more of it. More protein. Because we have more fluorescence with the actual antibody than we do with the isotype control. Okay. It's just sticking. It's just what I'm always minded of is. Do you remember as a child you might have had those little rubber octopuses that were sticky? You threw them on the window and they worked their way down. Now, when you drop them on the carpet, you pick them up. They've got all sorts of bits stuck to them. Okay. Now, if you had something that recognised the octopus shape, it would stick to the octopus. If you've got a little man that works his way down the window, the antibody wouldn't stick to the man because it's not octopus shaped. But if you drop it on the floor, it's still going to pick up bits of dust and fluff and so on. So, that's a good analogy. And I find it helps to actually use that sort of example because some of these concepts are quite alien if you're not, if you haven't been doing it yet. And that's the thing, cells, if you dropped a cell on the carpet, it pick up bits of fluff and so on. If you dropped it in a tray of antibodies, no matter what the antibodies get, something will stick. So
you say the isotopes
So the isotope, sorry, say that again?
the carpet.
Yeah, so the isotype is effectively the dust on the carpet. You're saying how much dust the carpet fluff is sticking to it. Because if a cell has 10 sticky spots, and it takes up 10 isotype controls, when you put it to your actual antibody, 10 of your actual antibody will stick to those sticky spots. But then you might have 40 of the protein, so you get another 40 binding to the protein itself. So you have 50 fluorescence with your antibody, and 10 with your isotype control, because you've got 10 sticky spots. We don't want them to bind to anything. That's the thing. The companies actually market them as. So when they sell a kit, they sell specific isotype controls. They're the same in every way. Same isotype, same animals, same fluorochrome, same chemistry. It's just that they won't bind to anything specific.
So I have left on your bench. So the three on the front will do the antibodies. Sorry, give me a second. So we have Christy. Okay, so I have left on the bench. So the first bench will do socks, like one at a time. Socks 2, on 4, SSA1, SSA4, and on the back we'll do all the all items. So just make sure.
Excellent, someone's been paying attention.
Yes, good. I was testing. So just make sure that you remove all the supernatant, okay? And get the 100 milliliters of wash buffer. You have also the wash buffer. I have one bottle to be shared on the front, and there's one bottle to be shared on the back. When you have done that suspension, then you need to add 10 microliters of each one of the antibodies to each sample. Okay, so each sample will have basically 4 antibodies.
Let's just pause, run, restart. So I'm just running the water through again. And we see some generally events in the bottom left corner. These are very, very small, very, very non-granular. So these effectively are debris, rubbish, bits of cells, flakes of plastic, whatever. There's a few carryover beads, giving us fluorescence. I'm not too worried about those. Now it's not very interesting. So what I'm going to do, I'm going to instead put on my QC beads. So these are polystyrene beads, and they have 8 peaks. So they have 8 different brightnesses of fluorochrome, and they have all fluorochromes. So they're what's called rainbow beads. They show up everywhere, in every channel. Now if I click on acquire, it's a few seconds just to prime it. Oh, it's lost all my settings. Why has it lost all my settings? There we go. Let's just readjust this up. Right. There we go. So when we run the sample through now with our QC beads on, we can see this population here. Okay? They're relatively small, they're somewhat granular, but if we look at our fluorescences, here we've got fluorescence 1, green, against fluorescence 2, orange. So the CLS versus P, effectively. And can you see the 8 different populations? And here, we have 3 against 4. Now, ideally, we want that to be linear. We're not getting an awful lot of linearity on channel 4. These red lasers are a bit flaky. The red laser, I think, is beginning to go. I think it's on its way out. I mean, it's fine, it'll work, but I wouldn't want to be using it for really critical experiments and really critical data. Yeah, I know. I mean, we'll get data, but it's just the sort of thing that, yeah. So we've got our 8 different populations. Now, at the moment, can you see there's sort of bits in between each population? That's because we're getting bits of debris and stuff in there as well. So what we can actually do is we can do what's called gating. So what I've done is on this one, I've drawn a region. Yeah, I've drawn a, can you see the little ellipse, little oval? It basically says we're not interested in the doublet beads, or where there's a few beads stuck together. We're not interested in the bits of beads or the debris or the rubbish. What we're interested in is just these single beads. So we've drawn that little ellipse around there, and that's why these are now in red, because they're only showing events that appear in that ellipse. Anything outside that ellipse that is not shown on here. So gating is really important, because when we're looking at ourselves, we don't want to look at our debris or our rubbish or our aggregates, we want to look at ourselves. Otherwise we're going to get an incorrect result. So this is basically cleaning up our data. So now when you look, you don't see much between the peaks. Now, what we can also do, we can view it as a histogram, and I'll just draw one on top for the moment. I can always get rid of it. Yeah, so these are QC beads. So these are just there to check how well the instrument is running. But they're quite good for looking at this stage. If I do an FL1, and then we do a gate. Yeah, well, it's going through and going to waste.
Okay.
So if we leave it on, eventually this tube will run out and it will dry out, and that's not good for the instrument. But we've got plenty in there at the moment, especially on low flow rate. So you can see these are 8 peak beads. 3, 4, 5, 6, 7. We've got 7 peaks. We've actually got some events. on the left-hand axis?
So you split it into four, right?
Well, you see, what we can actually do is, let's put that to 0.75, and then pause, restart. Okay. All the peaks have now moved up. They're all brighter. Well, they're not brighter per se. I've just turned up the sensitivity of the detector. So 1, 2, 3, 4, 5, 6, 7, 8 peaks. This instrument only has 4 log decades. Most of the ones are up to 7 or 8. So that's why we're sort of cramming them on a little bit. But what it does show is we can actually tweak our data. Can you see now our linearity has gone a little bit from here as well? But what it does show is our results are not absolute. So when we put our cells through, we cannot say they are fluorescent or they are not. You can't do that with a flow cytometer. Because you see this peak here, well, if I turned my detector up to 999 and restarted, that bottom peak is now probably here somewhere, or it might be here. Because I've just turned everything up, I've moved everything right along, so most of it's off the left-hand side. Now these beads are pretty much non-fluorescent, near enough, but they're showing as 10 to the 2. If I put this detector setting down to 150 minimum, and I pause and restart, I don't think we'll see anything at all. We're seeing some stuff on the edge, and that's it. That would say they're non-fluorescent. So we don't have an absolute measurement. The machine will not tell you, yes, they are fluorescent, or no, they are not. What it will do is tell you, yes, they are fluorescent compared to. So we might put through some cells that we haven't added antibody to. In our case, we can use isotype match controls, or whatever. But what you cannot do is you cannot say, yes, they are fluorescent. Now, obviously, if they've got loads and loads and loads of a really bright fluorochrome stuff to them, then even on minimum setting, we're going to have a peak here. And you can say that even if a minimum setting, they're obviously fluorescent. But people think you can put it on, it will tell you, are they fluorescent or not? No, it won't. It will tell you, are they fluorescent compared to. That's why we have to use appropriate controls. Okay? Well, so each of these peaks, if I put that back to about 580 and then pause and restart. So what's happening is each of the dots on here is an individual event, an individual bead. Each of those has a specific fluorescence in the other channels. So they're either bright 8, 7, 6, 5, 4, 3, 2, 1. The peak is the number of events in that peak. So what we can do is we can actually get some statistics. So we can put a little marker region on there. And we can say, right, identify just that peak. And what we can then do is we can show some stats. Unfortunately, you get these weird artifacts. Let me just enlarge that for a moment. So at the moment, we're looking at about 500 events per second. Okay? But of that brightest peak, there's around about 40 to 50 per second of the brightest population. That brightest population makes up 13% or so of all of the beads going through. So the ones we've identified here as beads, 13% or so are in that brightest peak. It's about what we expect. And their median fluorescence is about 4,300. So median is the middle event of a population. So it takes the middle of that peak, it says the middle of that peak is 4,300 or so. Now, at the moment, we won't get it. I'm just going to stop that. simply because I'm most of the way through that tube now. Let's put that up there. Trying to find enough room, that's why analysing it later is often easier. Right, let's just get rid of that one. Too many windows open, that's the problem. Now, what we can actually do then, we could put a marker region on here to look at this one's median fluorescence and this one. The median fluorescence is how bright that population is. The higher the number, the brighter the population is. The brighter it is, the more fluorochrome it has. The more fluorochrome it has, the more antibody it has. And the more antibody it has, the more of whatever it is the antibody sticks to is there, on or in that cell. Okay? So, if, for example, our cells came out here, they're going to be absolutely loaded with our protein of interest. If they come out here, there's a fair amount of it. Come out here, there's not a great deal. If they come out here, there's actually very, very little of our protein of interest on our cells. So in the same way as we can't tell whether something is fluorescent or not, what we cannot say is we cannot put a single tube on and say these cells have OCT4. We can say they have OCT4 compared to our cytometry control. Now, what we don't know is, does that mean they've got loads of it or a little of it? We don't know. So that's why we are using our control cell lines. We're using the biological control, the one that has our markers, using the cell line that doesn't have our markers. They effectively form our standards. Because if our test cell line, the one we're interested in looking at, has as much as our positive control, it's got quite a lot of it. If it has only slightly more than the negative control, it's barely expressing it, but there's a tiny bit there, and so on. So what we have to do, we have to use the location of our peaks, and as you'll see in your protocol, there's overlay histograms, so you've got one with another one over it. I will make use of those when I prepare the data for you. But make use of the stats. So what we know is that we could look at the stats of this region and see, right, what percent of everything going through our cytometer are beads? We can then say, well, what percentage of beads are in this high peak? So we can use stats that way to look at percentages, and we can then say how much fluorescence is there. Okay? Yeah.
I'll go back to the median.
Yeah.
So, does it take into account the population? So the shorter peak is on the left-hand side.
Yeah.
Might have less brightness, but has a bigger population.
Yeah, so the spread of it is called the CV, coefficient variation. That's how wide the peak is. It's the variability. The median is how bright the population is as a whole, and the percentage is also important. Because if we run it through, if we run your sample through, and actually we get 100 cells coming through, because for whatever reason you've aspirated your sample or you've lysed it or something like that, if we put it through and you only get 100 cells, the median is not going to be that informative because 100 cells isn't statistically significant. But we're going to run about 10,000 events from each tube. So you need to look at the number of cells there are, and the way I'll prepare your data is I'll show you the number of cells, and I will show you the amount of fluorescence on each as dot plots, like this one I had, the 2-colour dot plots. And I'll show you histograms for each of your markers with the median fluorescence.
There will be
Something like that.
that you have in your protocol, right? So you should have something. And after you just need to understand what each
Yep.
one means. So what would be the positives, the negatives, positive or negative in terms of
Yep.
graphics.
I may well also use quadrants. So what you can see on there is a quadrant where the plot is divided into 4. Not necessarily equal 4s, but 4. Generally, when people use quadrants, anything in the bottom left quadrant is a double negative. It has neither of those markers. This one and this one are single positives. It has that marker but not the other one, or that marker but not the other one. The ones in this quadrant are double positives. They have both markers. Now, what's important to remember is these ones here are also double positive. But on this plot, they would actually be in the negative quadrant because it's what I said about it being relative. So this one is double positive relative to that set of antibodies, etc., etc. Whereas this one is double positive because it sits up there. So the position of the quadrant has to be adjusted for individual experiments, antibody cells, etc. Okay. So I will give you the stats, and here we've got percentage, whatever that is, something 0.8 point, no 90. I can't read it.
But what's the difference for here, like double positive and
Yes. So you can see here there's only 1% double positive, and here it's whatever 90 something, 91%. So, well, I will do that for you. So what I'll do is I will take your data files away. I will send you back a sheet with your forward and side scatter, size and granularity, your fluorescence histograms with the quadrants on showing percentages, and histograms like this of all of your data. Okay, showing the median, so the amount of fluorescence there is. Okay.
And that would be we just make the analysis of that graph. So presenting that the graph is in your result section, describe the graph in the result section, and after discuss the data in the discussion. That was one of
Is that what I'm on? Right, so we now need to just finish the final few bits, then we can start running some samples. So the forward and side scatter are the size and the granularity readings. Yeah. You can look at it in all different ways.
When you're reading papers, how can you, and they're showing you,
we will go through it. We'll have a paper. We will have a
Yeah.
are they all keeping the same standards?
F for fat, S for spotting. Yeah, that's a good way of remembering it. Forward and side scatter, F is for fat, S is for spotting. Spotty. So you've got spotty cells, and you've got fat cells, and you've got fat spotty cells, and non-fat, non-spotty cells.
If the information also, the sources that they are quite surprised. There's explanation about all that, the explanation of all the way that the lessons go inside, and you can all post it in the slide, which is easier. So you can do those, you can support that information. Okay? So for now, so we can integrate the things that temperature, so following the equation, remove any tested volume by washing the cells. Need 2 milliliters of fertilization with water. Yes. So to define the cell color, we need to spend 400 micrometers. So what we need to do is basically add all the section so that it is enough. So then so we cannot hear it from we get just this 1 milliliter to be around so if we do it just with 1 mil, okay? So we spin.
But we can spin them first.
Yes. Okay.
Because what we don't want is the sheet, the fluid with our cells in. There's lots of free antibody floating in. And when the laser hits that cell, there'll be antibody around it, the fluid from. So you'll get a higher result. Now, sometimes in diagnostics, you do what's called a no-wash procedure. You just have to add antibodies around it, but you can't spin down. But as a rule, you're much better off spinning each time.
So, do you think it's going to spin there first, to wash spin again?
No, I would just spin once.
So we get now the 1 milliliter, and I to stop.
Yeah. So yeah, you add some extra, to just make to free up any because there's detergent, it really helps free any just generally stuck stuff on there, and just dilute down that antibody, and then you spin it. Because if the cells are a really high concentration of antibody around them, when you spin them, they actually catch antibody with them and push it to the bottom of the tube. Whereas if you put it in a bigger volume, then there's a bit more space and you don't catch antibody.
So if you were to spend like just 1 milliliter of the butter, okay, and that is living here, okay, so I think we have and just stop that. So don't take anything out at the moment.
Add 1 ml
Just
to each.
add 1 milliliter to each one of the tubes, okay, and change the tips every time, okay, for different quantities. And after adding that, we can transfer the tubes into the centrifuge.
might be worth putting in one of these at that point.
Yes, we can do it.
So what I'll do is I'll give you 3 of these each. You'll need to label them. Your 3 tubes. Yeah, they'll fit in those.
So we can have these ones. We can have the wall, right? So we can have these ones. So they're one, three, one, two, three,
They're a little bit. So once you've resuspended your pellet for 400 microliters, transfer it to that tube. Because that's what we'll use to run it now. Yes, that's good. Don't worry about using the filter. We've not got enough cells to have a lot. So, yeah, just don't worry about the filter. There shouldn't be any pumping at all from what's in it. So, you should be okay. The person who's going to get cross is me. I'm taking an executive decision. The problem is if we filter it and we lose some cells, which we might still do, because you do tend to get a little bit of fluid there just by certain section stuff. It doesn't all go through, and we lose half our cells. It makes it more difficult to run that. So if there's any pumps, then we can do it. So make sure you lay your three tubes. You might as well do that now while you're waiting for the centrifuge. Yeah, just write straight on the plastic. It's fine. And also make sure you put if you're the ones doing the isotype management control, please put IMC on all of your tubes. Because if not, we may end up getting confused about what's what. I mean, I know you're going to bring your tubes and so on, but if you're running the isotype management control, those are the ones we need to run first. That's great. So take not supinated, so at it, resuspending that before I have my properties, and then transferring it through to its appropriate
That's yes.
Yeah. So you can remove the leads from the tubes.
Yeah, it's not a fusion filter.
Just remove the leads, okay? Look up down and cells in.
They just pull off, they don't screw it.
And make sure that you let the cells to the right tube, okay?
Sorry? Yeah. Yeah, sorry.
Make sure
I think some of that, I think it's, I think it's just really boring. There's some top ones and little sorting top ones.
They're the ones I've been using, but for today I'll just. What was the difference between them?
We're doing each other cells then. Because when we get that PBS, we can straight transfer the sample into the new tube. So I will say aspirate first all the media, and after, when you have removed all the supernatant out, you then can add the PBS, resuspend the cells in the PBS, and transfer that into the new tube. Right? That's fine. So success is the next cell by the end of by the time that you run the suspension media goes like up.
And then hopefully we'll have something to see and have some nice data.
Yes, and after we'll have the levels.
Yep.
But the main one is to check yourself. So it gets as PBS, resuspend, and transfer. PBS, resuspend, and transfer. Okay. Format with one.
Yes, so you have to have PBS into there to resuspend the pipette, and then transfer.
Very careful, we will be difficult to take it out. Just try it out.
You can, yeah. You can click it up and down to resuspend if you want. You can click it to resuspend it and then use the same bit to transfer it to your system.
If you do it gently, you won't be inducing bubbles in the suspension. So if you do very gently and without adding air to the sample, the bubbles will be less probable to happen.
At least we're in biology, we're not doing chemistry. Chemistry is something about low. So you resuspend those. Yeah, I think that's resuspended. Yeah. Sorry, just put that up and down once or twice if you do so. Right. Doesn't matter if there are a few bubbles. It's not a major issue. It's important to get all your cells though. I would just put it back in your wrap, just in case your pen is still stuck or whatever, you can still come back. You throw it away. I mean, it may not do it, but it's always worth trying to put the tube on it up and down. It's always worth going back. Okay, well, what we'll do, we'll run them all at one go. Because obviously, we're doing group data. Yes, we need to go some match controls first, and I think it'd be good to run them in standard order, because renaming files on there is a pain. So we want to run file 1, 2, 3, 1, 2, 3, 4, 5, 4, 5, 6.
And you have all the 2, 3, 4 names. So, cell name, what we are,
Right, if everyone wants to then come round, we'll see what we've got. See, if I was feeling in a particularly jocular mood, I'd say, right, do we want to have a book on anyone not having any cells, or? But I don't think we will. It's a serious time at this. Everyone will have cells.
Don't breathe.
Okay. Let's move that across there. So I've been running it so far on setup mode. So setup mode is where you don't save any of the data. So all the debris and the beads I've been running through so far, I haven't saved any of that. So now we need to actually run a sample. So this is an isotype match control. Good. Move that to there. Start. Pause. I'm going to bring. Right, I have to do this reasonably quickly because it is still running sample through, and I want to have enough to run. Let's do that down to 500. So what I'm doing is I'm just adjusting the settings. Restart. I'm adjusting the settings so that my isotype match controls come out in the bottom left corner. Pause. I'm not going to run that one yet. Not save the data. Because what I need settings that will work for all three of my samples, because I can tweak them later. But what we don't want is we don't want them off scale anywhere. Good. Well, whoever the first person's tubes are, plenty of cells.
Yay!
Well done. I can't see which one that is on
I think
there.
it's Christy, I think.
Excellent. Good. Plenty of cells.
Christine's done this before though. So.
Ah, right. Experience, eh? Right. Cool. Okay, so that's fine for all of our samples, those settings. So I will use these settings now for all of our samples. So what we'll do, we'll first run the negative control cell with the isotype match control. So untick setup, so we click on acquire, and we should get 10,000 cells coming through. Or 10,000 events. Now what you can see is you've got a nice single population of forward and side scatter cells. Some cell lines grow in two different forms. So some cell lines grow where you get smaller and larger ones, or non-granular and granular ones, or whatever. What are we up to? We're running at a couple of 100,000 cells per second. I'm just going to leave them to run at that, because we're up to 6,000 already. And so this is the negative cells, and it's the first isotype match control. But I'll leave the tubes in the same order, so that we can see what order we've done them. So it stops. We can then put our next tube on.
And there's still liquid left.
And there is still liquid left. Yep, that's it. Which is good. Now these, you can see, this cell line, can you see the previous ones were nicely in that gate? This one comes down here. So these cells, many of them are smaller and less granular than the previous cell line. And that's not a problem. It's not a problem at all. All it means is that this cell line has a different size and granularity profile to the first cell line. But they're not the same cell type, are they? I can't remember what they are. So because they're not the same cell type, we wouldn't expect them to have the same size and granularity. Now we're running at 60 cells per second there. I'm just going to go up to medium to push up to about 100 cells per second. I don't want to go any faster than that. It's not optimal running it faster for various reasons. So I'll just push it through at medium. So we need to acquire 10,000 of these, then we'll acquire 10,000 of the actual cell line. But this doesn't look to be significantly different to that first cell line. Now, not looking different doesn't mean it isn't different. There might be differences in the median fluorescences. So I'm always very cautious when I say there's no difference. It doesn't look much different. And that's only our visual assessment. But that's fine because it looks like these cells have about the same general stickiness. Yeah. 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 So when it gets to 10,000, because that's my predetermined stop point, it will stop and save the file. Well, what it will do is it will go from, when it gets to the end, it's currently got a pause button which enables me to pause the acquisition. When it gets to the end, it'll actually stop, and that will go back to an acquire button again. Yes, and you can actually specify the prefix and the suffix and so on and so forth. Once it has done this one, and we're at just over 9,000 now, I'm just going to quickly double check the folder that it's putting the files in there, but it should be. Yes. So it's now gone back to the acquire button. If I bring up today's folder I made, we've got data 1 and data 2 in there, which is good because we've run 2 files. So then we put on number 3. And again, when I said about pattern recognition, about looking at shapes, what you want to be looking at is the forward and side scatter shapes. And where the red populations are. And again, these look to be in almost exactly the same place. So these cells look far closer in terms of forward and side scatter profile to the last one we did. They're different to the first one. Now, there's probably something you can infer from that. Is this positive cell line similar in cell type to the test cell line? They seem to have quite similar forward and side scatter profiles. That doesn't mean they're the same. It's a bit like if you're spotting animals in the wild. If you see it's got spots, it doesn't mean it's a leopard. But it shows similarities with leopards. Now, of course, if all the other profiles are the same for the different plurograms, so if it has spots and big teeth and claws and a long tail and it's cat-shaped, then it's a pretty good bet it's a leopard. Okay? Whereas if it's got spots, but it's actually got really heavy shoulders and really big jaws, it looks more like a dog, it's probably a hyena. And so it's all about looking at the similarities. And actually, it's not bad analogy. I have to remember that one for future use. Because that's sort of what it comes down to. Because what we don't know is, we don't know what these cells are. Well, obviously, some of us do, but you don't. So you have to derive all of your information based upon what you get from the flow cytometer. Now, what you're not getting is a little label popping up saying these are fibroblasts, or these are epithelial cells, or these are lymphocytes. You haven't got that. You can only extrapolate that data from the information you get. But there is quite a marked difference in forward and side scatter between the first sample and the other 2. So that alone tells you something. Right, well, number 2 has got really good cell numbers. Because that's only taken about 40 seconds. Who was person number 2? Excellent. Have a metaphorical gold star. Good numbers going through. See, that was 43 seconds. Can we beat that, do we think? Anyone beat that? That's a bit of a challenge. We won't do absolute cell counts or anything like that, but so that's pretty good as well. But also bear in mind that different cells will behave differently when you label them. So some cells, there's one cell I call QVEC that I used to use, a lung cell line. Even if you centrifuge it on the lowest settings, these cells just fall apart. Have you ever used QVEC? They're awful. Basically, you pick up the flask and half of them have died. You know, they're so fragile. Whereas I've also used Hackett cells, keratinocyte cell line. Now, normally when you trypsinase cells, you wash the cells off, you put your trypsin in, you wait for a minute, you tap your flask, they're all coming off, you add your media to neutralise and so on. Hackett cells, you take off the media, you put on some PBS EDTA and wash them, and do that again. Then put your trypsin on, put them in the incubator for 15 minutes, take them out, they're not coming off yet. So you take off your trypsin, you put fresh trypsin on, you then put it back in the incubator for another 10 minutes, and then you get your flask out and you go bang, 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 bang on the desk, and they start to come off. Yeah, and I've had trouble scraping them. So you're going like this, and they're just not being scraped off. So they Hackett stick like, it's like they're welded to the plate. And in fact, once I was banging the flask so hard on the desk that the neck snapped off and flew across the cell culture. And I thought, well, they're not sterile anymore, are they? So they're awful. But with Hackett, no, don't try not to break fast. But with Hackett, when you spin them down, you cannot kill them. You cannot break them up because they are just rock solid. So if you're doing an experiment with QVEC and Hackett, you cannot have the same protocol for both, like we did here. Because the Hackett will be fine and the QVEC will all just be shredded. But if you do it so low to preserve the QVEC, you won't get your Hackett. So although we've got sort of a standardisation of protocol, what we see in one cell line is not what we'll see in the other one, even for things like cell counts. So, but that was good. That was 2 minutes. That one. This looks to be fairly rapid. 1,000 there. Good. And so this is the last of our isotype match controls. So we've run them in duplicate. And again, that's quite important to run in duplicate because it only takes someone to accidentally put their pipette into the wrong antibody tube and put a little bit of antibody into the isotype match control and it's wrong. So running it duplicate means when I look at this data later, I can see that we've got 2 isotype match controls that are the same for each of our cell lines. Yeah, we're up to about 5,000, so we're almost there. And then our actual samples. So far, no one's lost their pellets, no one's lost their cells. So brilliant, those 2. Now, of course, the other 3 of you may or may not have cells, but we should have. From what I could see, everyone had good technique, so that's fine. And actually, in some ways, you're better at it when you're new at it because you're more careful. When you've been doing it a lot, you end up going and when you've got 40 tubes and you've got aspirate, you end up rushing. Well, yeah, I've done that. I've done that when I've been in a hurry. I've actually, spun them down, poured the supernatant off, given it a quick tap to get rid of it, added my antibody, I re-spent it, put it back in, just because I'm in a hurry. Now, is it optimal? No, not really. But again, sometimes and I'm not recommending that for a moment. Right. It's all about adapting. You have to do what you have to do sometimes. And it may not be optimal, but sometimes you have to just because you have no choice. You can get multi-channel pipetters, but I was talking to someone the other day, and they were actually, when they were running repeats of something, they were getting a curve. Their results. Turns out the multi-channel pipetter wasn't doing the same volume. When they took up, say, 100 into it, you look down the line of tips. It was like that rather than flat across. Just be careful with multi-channel pipetters.
It's 12 o'clock now. My I don't know how long with time.
We've got fluorescence. I mean, the thing is, I'm happy to just keep. I'll keep running these anyway. I suppose it depends. If you have other classes you have to get
that's
to.
What I'm saying. So if you have time to stay, you want to stay on the ledge and everything is running, that's fine. If you have things that you need to attend after, you can leave now, and what we will do is we share the data. We will have time to have another discussion about these data collection. But so you will have access to the data. Okay, so yes, this is for your season, first part of your summative work.
And I'll put together a one-page handout on just roughly how it works and stuff as a summary.
Yes, so but if you are okay to stay in for a bit longer to finish it, yeah, that's all it is. Yeah, okay.
Right. Whoa. Up in there. I have no idea what that was. Oh, nice fluorescence there. It's much stronger. So tube 2 is stronger than tube 1 in all 4 markers, it would appear. Actually, the thing is, the mask, perfect poker face, isn't it? Is it supposed to be that? Whereas, you see, if you look round and you see a panicky look, it's like, it's not worked. Or if you see a smug look, it's, yeah, perfect. But yeah, you can't tell with the mask. So.
In those ones, they said the second one, so those ones are the share system.
Yeah.
Yeah, so those ones are the positive.
Right. So the positive control is brighter than the negative control, which is good. It's good. There are experiments I've run and I've seen where that isn't the case. And then you've got to try and work out what's going on. But here, that's good. We've got staining where we should have staining and not where we shouldn't. So far. So it's worked really nicely so far. And then the interesting one will be the first of the test
The
cells.
share 6 are all positive control. And yes, they are the negative control. In your report, you will need to speak about the controls, okay? They are quite important. You need to speak in terms of internal controls and external controls. So you will need to understand what are the main markers that should be present for stem cells, okay? So which ones you would expect to see and which ones you shouldn't see, which ones can you use as a negative to say, okay, this marker can't be there. So if this marker is not there, then we can together with our positive controls, we can say that actually the cells are in a pluripotent stage. There are markers that you see as actually,
Oh, yes, fantastic. My gut feeling is not quite as bright as the positive controls, but definitely positive. This is the third of the set.
Okay, yes, this is the ending. Have we done the negative one yet?
Yep. Negative had very little in it. Positive had quite a lot. This has got some. Again, the actual numbers will reveal the. And what I would say is, if you're running flow cytometry, try not to get too excited by what you see on the screen, because when you come and analyse it and you apply the gate properly, bear in mind we haven't got a proper gate up here at the moment. So we're not. Our red plots are not absolutely representative of what our data will be. It's very much a quick look. We're quickly assessing our data.
So when you take those, then you will need to analyze, and you're going to be all about characterization of your cell line, of the N8 cells. Okay? To characterize, you want to understand ideally, so we will know that cell line should be on a pluripotent, so we use pluripotent stem cells, so it should be in a pluripotent state. So what we're trying to see is characterize the cell line. Actually it is on that site. Okay, so that around about the good bodies themselves, speak about the markers that should be positive, that should be negative, and after speak about the controls that you use, the techniques that you are using, and there will be different techniques to do characterization, the technique itself, and analyze actually the data. We need to maybe troubleshoot a little bit if something doesn't go as you might think that should have gone. Just troubleshooting and suggest future things that can be done to better characterize the data. Okay, so that's the thing. You will see that I left that's a research paper just out of curiosity where they do the characterization also of in this one of the cell line that they were trying to produce. So they use different techniques, including this one to do the characterization. So there is a little bit about it. And they have included some of the graphs on their data. So you can also see how to present the data, how to label the images, even kind of a little bit of legends and things. So you can find any of these sessions as an example. And you will need to find more in terms of how to advise people more in these ones, you will see different ways of presenting the data. This is just one of them. This is our fluorescence images that we will be doing next week.
But the labelling process for microscopy is pretty much the same. You could, in theory, take these tubes, put some of it on a slide and look down. What you don't see, though, is you don't see morphology. Because they've been taken off their substrate, they're all rounded up. So they won't be spread out like a growing cell will.
Yes, so that's why in here, in this paper, for example, they are combining the immunofluorescence, where they see the cells, they stain the cells that are attached still to the surface, so to the growing plate. Right. So they need to detach them. So you can see the morphology of the colony, how the cells spread, you can see exactly how they are, right? And that's what they do the staining. You can also detach them and splash them on a slide and do the same procedure. You move a little bit in terms of the morphology of the cell itself, the columns, therefore the columns or even the cell morphology because they become forms. Okay, but you still get the labeling done because you have the cells, even if they are suspension like these ones, so you still have everything attaching to it. It's just in terms of the morphology that you would usually get. But that is something that you after can discuss when you do your reports. There are things that you will for this report. Now for this one, we are in this one. So this one is quite good report in terms of the use of the book. This one is genotype. So they did different techniques to characterize the cell line. So we will be doing two techniques. We'll be doing this one and we'll be doing the immunofluorescence later. And now we combine both and we explore to characterize the cell lines. Because we will visualize also the cells in growing in the previous image as in the cell culture. Yes. Question more. So if you want from those, you can even take some pictures of those, and after you can include that picture as a base on your report to see the cells growing on a plate culture, and after the cells being produced. They do like freeze. They're like freeze. So we're doing some of them, yes, we will be using probably the photos to about yes, okay. So that is a little bit, so the things that end up embedded into the model in some way. So you just need to keep that in mind and use that information for when you are writing the report itself. Okay. How long is there?
The final one, tube one, the final person, tube one went through in 31 seconds.
Ooh,
So,
good.
good cell retention there.
Oh. So we have a winner then. We have first place and second place. Okay, that's good.
But what I would say is everyone's had really good cell numbers.
That's good.
No one has sort of butchered their pellets, as in no one sort of, we're having to wait 10 minutes to just about scrape a few thousand cells. It does happen sometimes. I've done experiments where I've had to. You start with low numbers and you lose a lot, and you click on acquire, and you sit there for literally 25 minutes waiting for a tube to run down, and you just about get maybe 2,000 cells. We've not had that. All of our cells have gone through at a decent rate. You've not lost huge numbers. How many did we start with per tube?
5,000? No, 50,000.
You started with 50,000. Well, also, you see, starting with 50,000, 50,000 is actually quite a tough number to start with as well. 500,000? We see a routine standard number that many people would start with would be 1 million. Well, that's still half what would be considered a normal standard number of cells to start with. So you've done really, really well. Because the more cells you have, the bigger the pellet. The bigger the pellet, the easier it is to avoid taking cells. You often start with 1 million cells, yeah. But then you see what you could be looking at is you could be looking at 100,000 events, but then you might be looking at subpopulations within there. So you might need to acquire 200,000, 300,000. So you start with 1 million to ensure you've got more than enough. But yeah, 1 million is reasonably standard. So you started with a fairly small pellet, and you've not lost that many.
Well done.
So yeah, give yourselves a clap actually, because it's the first time you do that, and we're up to 9,800 on the last tube, 10,000. There we go. That's it. They're all run.
Fantastic.
So do we know the? You made a note of the order of the people, haven't you? What order they were in?
I will just take a picture of that and I will write it.
Yeah, so that we know who's run and we know in what order we've run them. So we've now got.
You will be able to get all we will put all the data available, so you can use, if you want, you can compare, because we have repeats, so
Yep.
you can compare the data of the repeats if you want. from
Yes.
the report.
So we've got 15 data files. So I will take those away, I'll process them all, I'll turn them into plots and charts and histograms and stats. I will send them back, and once they're perfect, then they can be sent out to everyone for you to analyse. And I'll send them hand out as well.
And after we will kind of do a session just to have the discussion of the data, okay, when we have this.
Yeah.
We will be sent the data, we'll have a look at it, and after that we'll do the session just for answering questions.
Do a remote session,
remote
yeah.
session online, yeah, okay. So we do that. And